This is chapter 16. We're going to look at the endocrine system. So the nervous and endocrine systems act together to coordinate functions of all body systems. Now remember that the nervous system acts through nerve impulses, also known as action potentials, and these are conducted along the axons of neurons. At synapses, nerve impulses will trigger the release of mediator messenger molecules, which in the nervous system are known as neurotransmitters. Now, the endocrine system also controls body activities by releasing mediator molecules, but in the endocrine system, these are called hormones. The means of control of the two systems are very different, though. Now, a hormone is a mediator molecule that is released in one part of the body, but it regulates the activity of cells in other parts of the body, often from very far distances from where it's released to its target cell. And this is because hormones circulate through the blood. Now, both neurotransmitters and hormones exert their effects by binding to receptors on their own target cells. Now, this table here compares the characteristics of the nervous and endocrine systems. Now, in this chapter, we will be focusing on the major endocrine glands and hormone-producing tissues, as well as examine how their hormones govern body activities. Now remember that the body has two types of glands: exocrine glands and endocrine glands. Now first on this slide here we have the exocrine glands. Remember, these are glands that secrete their products into ducts that carry the secretions into body cavities or into the lumen of an organ or onto the outer surface of the body. So exocrine glands include sudoriferous glands, sebaceous glands, mucus glands, and digestive glands. Now, endocrine glands secrete hormones. So they will secrete their products, which are hormones, into the interstitial fluid surrounding the secretory cells rather than secreting their products into ducts. Now, from the interstitial fluid, the hormones will diffuse into the bloodstream by going into blood capillaries, and then the blood will carry those hormones to target cells throughout the body. Now, because of their dependence on the cardiovascular system to distribute their product, endocrine glands are some of the most vascular tissues in the body. So this means these endocrine glands have a lot of blood vessels associated with them. Considering that most hormones are required in very small amounts, circulating levels typically are quite low. Now, the endocrine glands include the pituitary gland, the thyroid gland, the parathyroid gland, adrenal gland, and the pineal glands. Now, in addition, there are several organs and tissues that are not exclusively classified as endocrine glands, but they do contain cells that secrete hormones. So these include the hypothalamus, the thymus, the pancreas, ovaries, testes, kidneys, stomach, liver, small intestine, skin, heart, adipose tissue, and the placenta. So taken together, all the endocrine glands and hormone secreting cells will constitute the entire endocrine system. Now, the science of the structure and function of endocrine glands and the diagnosis and treatment of disorders of the endocrine system is known as endocrinology. Now, there are four main functions of hormones. We will go over them here. Our first one is to help regulate. So we're going to regulate things like the chemical composition and volume of the internal environment, so the interstitial fluid. So we'll just say composition. So this is the chemical composition. And volume of the interstitial fluid. I'll just put IF. That stands for interstitial fluid. They also help to regulate the metabolism and energy balance. They help to regulate the contraction of smooth and cardiac muscle fibers. They help to regulate some glandular secretions. Glandular. And they also help to regulate some immune system activities too. Okay, our second major function, so those all kind of fall under regulation. Our second one is to control growth and development. Our third overall function of hormones is to regulate the operation of reproductive systems. And our fourth function would be to help establish circadian rhythms. Okay, so here in this picture, we have some of the glands. So we're looking at the main endocrine glands being the pituitary gland, the thyroid, the parathyroid, which is going to be behind the thyroid glands there, the adrenal glands on top of the kidneys, and the pineal gland. So those are our endocrine glands. And then our organs and tissues that are not classified as endocrine glands, but they do contain cells that secrete hormones. These would be like the hypothalamus, the thymus, the pancreas, the ovaries and testes, the kidneys, the stomach, the liver, the small intestine, heart, and then also the skin, adipose tissue, which is fatty tissue, and the placenta would also be included in those organs that are not endocrine glands but still secrete hormones. Now, even though a given hormone travels throughout the body in the blood, it affects only specific target cells. Now, hormones, just like neurotransmitters, influence their target cells by chemically binding to specific protein receptors. Now, only the target cells for a given hormone have receptors that can bind to and recognize that hormone. Now, receptors, like other cellular proteins, are constantly being synthesized and broken down. And generally, a target cell has 2,000 up to even 100,000 receptors for a particular hormone. Now, if a hormone is present in excess, the number of target cell receptors may decrease. This is an effect called downregulation. For example, when certain cells of the testes are exposed to really high concentration of luteinizing hormone, the number of luteinizing hormone receptor decreases. So downregulation makes a target cell less sensitive to a hormone. So this is when a hormone is present in excessive amounts. We don't want the target cell to be that sensitive to excessive amounts of hormone. So again, downregulation makes a target cell less sensitive to a hormone. Now, in contrast, when a hormone is deficient, the number of receptors may increase. This phenomenon is known as upregulation. Now, upregulation makes a target cell more sensitive to a hormone. So again, if there is very little hormone present, upregulation makes a target cell more sensitive to that hormone. Most endocrine hormones are circulating hormones. So what this means is that they pass from the secretory cell that makes them into the interstitial fluid around the cell, and then into the blood. And then they will travel a distance in the bloodstream. Then they will leave the blood, go back into the interstitial fluid, and then act upon their target cells that are usually a distance from the original endocrine cell that secreted them. So these are circulating hormones. Now, other hormones are called local hormones, and they will act locally on neighboring cells, or even they'll act on the same cell that secreted them without first entering the bloodstream. So local hormones that act on neighboring cells are called paracrine hormones. And hormones that act on the same cell that secreted them are called autocrine hormones. Now, local hormones are usually inactivated very quickly, whereas circulating hormones may linger in the blood and exert their effects for a few minutes or occasionally a few hours. In time, the circulating hormones are inactivated by the liver and then excreted by the kidneys. Chemically, hormones can be divided into two broad classes: those that are soluble in lipids and those that are soluble in water. Now, this chemical classification is also really useful functionally because the two classes exert their effects differently. The lipid-soluble hormones, these include the steroid hormones, thyroid hormones, and even nitric oxide. Now, these hormones are only able to circulate in the blood attached to transport proteins. Because they're not soluble in the watery fluid of blood, they're not able to travel in the blood alone. Water-soluble hormones, these include things like amine hormones, peptide and protein hormones, and eicosanoid hormones. These are able to circulate freely in the blood plasma. And this is because they're able to be dissolved in the watery blood, since they are water soluble. Now, these are the three types of the lipid-soluble hormones. Up top here we have the steroid hormones. These are going to be derived from cholesterol. You can kind of see that they look very similar to cholesterol if you remember that 4-ring structure of cholesterol. So they look very similar. And there's going to be various sites on these 4 rings for different chemical groups to attach to. These small differences allow for a large diversity in function. Next, we have the thyroid hormones. So this is T3 and T4, our 2 thyroid hormones. And they are synthesized by attaching iodine. See these iodine molecules here onto the amino acid tyrosine. Now, the presence of these 2 benzene rings, these purple rings here, this makes these molecules very lipid soluble. So they're very hydrophobic. Then we have the gas nitric oxide. This is both a hormone and a neurotransmitter, and its synthesis is catalyzed by the enzyme nitric oxide synthase. So like we said earlier, the water-soluble hormones can travel freely in the blood, so they don't have to be attached to anything. But most lipid-soluble hormone molecules have to be bound to transport proteins. Now, these transport proteins are synthesized in the liver, and they have 3 general functions. The first one is that they're able to make lipid-soluble hormones temporarily water-soluble. So this increases their solubility in blood. They also reduce the passage of these small hormone molecules through the filtering mechanism in the kidney. So by doing so, they are slowing the rate of hormone loss in the urine. So they will reduce the lipid-soluble hormone loss in the liver. Or sorry, through the liver in the urine. Sorry, that's through the kidneys. Goodness. And they also provide a ready reserve of hormone that's already present in the bloodstream. So they're just kind of freely floating, just ready to be available to be used when that hormone that they are attached to is needed. So kind of a reserve of hormone. Now there are also 3 types of the water-soluble hormones. So we have the amine hormones. These are synthesized by decarboxylating amino acids. So decarboxylating, we talked about it a little bit before, but decarboxylating means to remove a molecule of CO2. And they are called amine groups or amine hormones because they retain an amino group. Peptide hormones, these are amino acid polymers. So these smaller peptide hormones, these consist of chains to like 3 to 49 amino acids. And then the larger protein hormones consist of chains up to 200 amino acids, give or take. And then we have the eicosanoids. These are derived from a 20 carbon fatty acid chain. And the 2 major types of eicosanoids are prostaglandins and leukotrienes. Now, most water-soluble hormone molecules will circulate in the watery blood plasma in a free form, which essentially just means that they're not attached to other molecules. Now, the response to a hormone depends both on the hormone itself and also the target cell. Various target cells respond differently to the same hormone. So the response of a hormone is not always the synthesis of new molecules, as is the case for insulin, but there could be other hormonal effects, including changing the permeability of the plasma membrane, stimulating transport of a substance into or out of the cell, altering the rate of specific metabolic reactions, or causing contraction of smooth muscle or cardiac muscle. Now, in part, these varied effects of hormones are possible because a single hormone can set in motion several different cellular responses. However, a hormone must first kind of announce its arrival to a target cell by binding to its receptors. Now, for lipid-soluble hormones, their receptors are located inside target cells. The receptors for water-soluble hormones, on the other hand, these are going to be found in the plasma membrane of the target cells. So here we can see the lipid-soluble hormones. So as we kind of just learned, the lipid-soluble hormones, including the steroid hormones and thyroid hormones, will bind to receptors inside the target cells. So first off, we can see these hormones traveling through the blood attached to a transport protein. The transport protein will bring them to the target cell, and the lipid-soluble hormone can directly just diffuse from the blood through the interstitial fluid and then through the lipid bilayer of the plasma membrane into the cell. Because it's lipid soluble, it can very easily diffuse through the lipid bilayer because it's also a lipid. Now, if the cell is a target cell, the hormone will bind to and activate receptors located within the cytosol or sometimes within the nucleus. So here we have a receptor for this lipid-soluble hormone found within the nucleus. And the hormone will bind to the receptor, and it's going to have an activated receptor hormone complex. And this activated receptor hormone complex will then alter gene expression. So it can actually turn specific genes of the nuclear DNA on or off. So as the DNA is transcribed, a new messenger RNA will form. It will leave the nucleus and enter into the cytosol. And there it directs the synthesis of a new protein. So we'll remember how the messenger RNA will bind to a ribosome, and this is where transcription takes place. That mRNA directs the synthesis of specific proteins on the ribosomes. Then this new protein can alter the cell's activity and cause responses typical of that hormone. So on the right picture, essentially it is the same thing occurring, but here the receptor protein is found within the cytosol instead of the nucleus. So both of these can occur with lipid-soluble hormones. Now, because amine, peptide, protein, and eicosanoid hormones are not lipid-soluble, they can't diffuse through the lipid bilayer of the plasma membrane and bind to receptors inside target cells. Instead, water-soluble hormones will bind to receptors that protrude from the target cell surface. When a water-soluble hormone binds to its receptor at the outer surface of the plasma membrane, it acts as the first messenger. So the first messenger, the hormone, then causes production of a second messenger inside the cell where specific hormone stimulator responses take place. Now, a really common second messenger is cyclic AMP or CAMP. So when we're looking at the action of a typical water-soluble hormone, it happens like this. So we can see that these hormones can travel freely in the blood. They will diffuse from the blood through the interstitial fluid and then bind to its receptor at the exterior surface of the target cell's plasma membrane. So you can see this binding here. Now we have this hormone receptor complex, and it activates a membrane protein called a G protein, this little blue kidney-shaped protein here. Now the activated G protein will in turn activate adenylate cyclase, and adenylate cyclase converts ATP into cyclic AMP. Now, because this enzyme, the adenylate cyclase, has its active site on the inner surface of the plasma membrane, this reaction is all going to occur within the cytosol of the cell. So cyclic AMP acts as the second messenger, and it will activate one or more protein kinase. Now, protein kinases are usually free in the cytosol, and protein kinase is an enzyme that phosphorylates other cellular proteins. So remember, phosphorylates means that it will add phosphate groups. Now, the donor of these phosphate groups is usually ATP, which is converted to ADP. So once we activate those protein kinases, they will add phosphate groups, as you can see down here, to other proteins. Taking that phosphate group from ATP, converting it to ADP, and then adding that phosphate to the protein. Now, phosphorylation kind of activates some of these proteins, and it kind of acts as an inactivation to other proteins. So essentially, phosphorylation kind of acts like a switch, turning on or turning off other proteins. Now, these proteins, they could be enzymes, or they could be other cellular proteins. There's lots of different proteins in the cell that can be phosphorylated by protein kinases. Now, once we have these phosphorylated proteins, these will in turn cause reactions that produce physiological responses. Now, different protein kinases will exist within different target cells and even within different organelles in the same target cell. So one protein kinase may trigger glycogen synthesis, another protein kinase may break down triglycerides, a third might promote protein synthesis, the list goes on and on. So this pathway is very specific, but there's lots of different things that could happen. So one specific water-soluble hormone could activate a protein kinase that specifically triggers glycogen synthesis. That's its job, and that water-soluble hormone is all that it'll do. But another type of water-soluble hormone may activate protein kinases that do something completely different. So it all depends on the hormone. Now, after a brief period, an enzyme called phosphodiesterase up here is actually going to inactivate cyclic AMP. So in this way, a cell's response is turned off unless a new hormone molecule binds to the receptor in the plasma membrane. So it's kind of a short-lived response. It's not going to continue forever. Now, hormones that bind to plasma membrane receptors can induce their effects at really low concentrations, and this is because they kind of initiate a cascade or kind of a chain reaction. Each step can multiply or amplify the initial effect. So this is because the binding of the hormone can activate very many G protein. We could even have like hundreds of G proteins activated, and each of those G proteins could activate 100 adenylate cyclases, which would give us 100 cyclic AMP per G protein. And then each of those cyclic AMP could activate another 100 activated protein kinases. So by the end, we have like, I don't know, say like 100 million of these phosphorylated proteins, all from one hormone receptor activation. So it's really a cascade amplification effect. So that's why these hormones that bind to plasma membrane receptors really have to be at low concentrations because of this huge amplification that occurs. The responsiveness of a target cell to a hormone depends on a couple things. First, it depends on the hormone's concentration in the blood. It also depends on the abundance of the target cell's hormone receptors for that specific hormone. And it also depends on influence exerted by other hormones. So other hormones can actually have an effect on how a target cell responds to a different hormone. Now, when the effect of two hormones acting together is greater or more extensive than the effect of each hormone acting alone, the two hormones are said to have a synergistic effect. When one hormone opposes the actions of another hormone, these two hormones are said to have an antagonistic effect. The release of most hormones occurs in short bursts, with little to no secretion between these bursts. Now, when stimulated, an endocrine gland will release its hormone in more frequent bursts, increasing the concentration of that hormone in the blood. In the absence of stimulation, the blood level of the hormone will decrease. Now, regulation of secretion normally prevents overproduction or underproduction of any given hormone, and this works to help maintain homeostasis. Now, hormone secretion is regulated by signals from the nervous system, by chemical changes in the blood, and even by other hormones. For example, nerve impulses in the adrenal medulla will regulate the release of epinephrine. blood calcium level will regulate the secretion of parathyroid hormone, and a hormone from the anterior pituitary gland, which is the adrenocorticotropic hormone, stimulates the release of cortisol by the adrenal cortex. Now, these are all examples of hormone regulation that we will go over individually at later points in this chapter, but these are just examples of how hormone secretion is regulated. Now, most hormonal regulatory systems work by a negative feedback mechanism. So this diagram here, just like this one, it shows blood pressure. So when blood pressure is increased, it stimulates the release of hormones that dilate blood vessels to decrease blood pressure. And so this is a negative feedback loop to return homeostasis. Now this is a negative feedback loop that we've seen before, where the baroreceptors detect that increase in blood pressure, and hormones are actually going to be released to dilate those blood vessels. There are a few hormones that operate by a positive feedback mechanism. Again, we have also seen this positive feedback loop too for oxytocin. So this is during childbirth, where there's increasing stretching of the cervix, and this is detected by stretch receptors in the cervix, and the brain interprets this and releases oxytocin. Now, oxytocin will actually cause the walls of the uterus to contract more forcefully, causing the baby to be pushed out further and causing more stretching of the cervix, and then more oxytocin is released, and it's kind of this large snowball effect, which is the positive feedback loop. So that's an example of positive feedback in hormone secretion. Now, for many years, the pituitary gland was actually kind of called the master endocrine gland because it does secrete several hormones that control other endocrine glands. Now, we now know from research and experimentation that the pituitary gland itself has its own master, which is the hypothalamus. Now, this small region of the brain below the thalamus is a really major link between the nervous system and the endocrine system. Cells in the hypothalamus synthesize at least 9 different hormones, and the pituitary gland secretes 7. The pituitary gland lies in the hypophyseal fossa of the sphenoid bone, and it attaches to the hypothalamus by this stalk called the infundibulum. So here we can actually see the whole pituitary gland. The pituitary gland has 2 anatomically and also functionally separate portions. These are the anterior pituitary and the posterior pituitary. As you can see here, the top portion of the pituitary gland is the infundibulum, which is going to attach it up here to the hypothalamus. So the anterior lobe, or the anterior pituitary, which is also known as the adenohypophysis, this accounts for about 75% of the total weight of this gland, and it's composed of epithelial tissue. The adenohypophysis secretes 7 hormones. We will look at those specific hormones yet. Now, the posterior pituitary, or the posterior lobe, is also known as the neurohypophysis, and it's actually composed of neural tissue. Now, it actually doesn't create any hormones itself, but it does release two hormones that are made by the hypothalamus. Now, the anterior pituitary secretes hormones that regulate a wide range of bodily activities, from growth to reproduction. Now, release of the anterior pituitary hormones is stimulated by releasing hormones, and it's suppressed by inhibiting hormones that both come from the hypothalamus. So the hypothalamus will control what hormones are secreted by the adenohypophysis, and it does so by secreting these releasing hormones or by secreting inhibiting hormones that control the adenohypophysis. Now, in this way, the hypothalamic hormones are a really important link between the nervous and the endocrine systems. So here we can see the hypothalamus here. Now, these releasing and inhibiting hormones synthesized by the hypothalamus are actually specifically synthesized by hypothalamic neurosecretory cells within the hypothalamus. So these hormones are created by these neurosecretory cells and then transported within axons and released at the axon terminals. Now, these hormones will actually diffuse into capillaries of the primary plexus of the hypophyseal portal system. And then they're carried by the hypophyseal portal veins to the secondary plexus of the hypophyseal portal system, and this is for distribution to target cells in the anterior pituitary. So here we have that primary plexus of the hypophyseal portal system. They will travel along these hypophyseal portal veins, and then they will be carried to the secondary plexus of the hypophyseal portal system, where they're going to be distributed to those target cells in the anterior pituitary. Now, this portal system is essentially just a net of capillaries. So in a portal system, blood is going to flow from one capillary network into a portal vein and then into a second capillary network before returning to the heart. So we kind of see that here. We can see this first capillary network flowing down with a vein to a second capillary network. So essentially this is how a portal system works. Two capillary networks connected by a vein, a portal vein, before returning to the blood, or returning to the heart, after that second capillary network. Now, the name of a portal system indicates the location of the second capillary network. So in the hypophyseal portal system, the blood flows from the capillaries in the hypothalamus to portal veins that carry blood to the capillaries of the anterior pituitary, also known as the adenohypophysis. So very quickly, those clusters of specialized neurons called the neurosecretory cells, these are found above the optic chiasm, and they synthesize the hypothalamic releasing and inhibiting hormones in their cell bodies, and then they package the hormones inside vesicles, which then are transported along the axon to the terminals. So just kind of specifying what those neurosecretory cells are. They are neurons, but they create hormones. So let's quickly look at some of the hormones of the anterior pituitary gland. First off, there are 5 types of anterior pituitary cells. So these are somatotrophs, thyrotrophs, gonadotrophs, lactrophs, and corticotrophs. So these are going to be our 5 types of cells within the anterior pituitary. We can see that these somatotrophs secrete human growth hormone. This is also known as somatotropin. Human growth hormone does a lot of things, but one thing it does is, in turn, it stimulates several tissues to secrete insulin-like growth factors. So human growth hormone itself can stimulate other tissues to secrete. So insulin-like growth factors, often we just call these IGFs. You'll see this come up again later. But those insulin-like growth factors are hormones that stimulate general body growth and also regulate aspects of metabolism. So IGFs are really important hormones, and the human growth hormone stimulates other tissues to secrete IGFs. Thyrotrophs, these secrete the thyroid stimulating hormone, or TSH. TSH controls the secretions and other activities of the thyroid gland. So it kind of makes sense. It's the thyroid stimulating hormone. It's going to control the thyroid gland. Now, gonadotrophs, these secrete gonadotropins. Now, 2 gonadotropins are the follicle stimulating hormone and the luteinizing hormone. FSH and LH both act upon the gonads. They stimulate secretion of estrogens and progesterone and the maturation of oocytes in the ovaries. And these gonadotropins will also stimulate sperm production and the secretion of testosterone in the testes. Lactotrophs secrete prolactin, PRL. This is a hormone that initiates milk production in the mammary glands. And then corticotrophs secrete adrenocorticotropic hormone, or ACTH. This stimulates the adrenal cortex to secrete glucocorticoids, like cortisol. So that's a really important one. Corticotrophs will also secrete melanocyte-stimulating hormone, MSH. We won't much talk about the role of MSH in this class, though. Secretion of the anterior pituitary hormones is regulated in 2 ways. So the first way is that the neurosecretory cells in the hypothalamus secrete 5 releasing hormones, which stimulate the secretion of anterior pituitary hormones, and 2 inhibiting hormones, which secrete the suppression of anterior pituitary hormones. So this is going to be our first regulation of anterior pituitary secretion. So the first one is by the releasing hormones and inhibiting hormones. From the neurosecretory cells in the hypothalamus. And then the second way is negative feedback in the form of hormones released by target cells decreases the secretion of 3 types of anterior pituitary cells. So our second way is by negative feedback. Now, in these negative feedback loops, the secretory activity of the thyrotrophs, gonadotrophs, and corticotrophs decreases when blood levels of their target cell gland hormones rises. So those are going to be our 3 cells that we can control with negative feedback loops. Again, that's the thyrotrophs, gonadotrophs, and corticotrophs. So for example, we have on this picture on the left here an example of a negative feedback loop with the anterior pituitary gland. So here we have the adrenocorticotropic hormone, also known as corticotropin, going to be released from the anterior pituitary gland. It's going to stimulate the cortex of the adrenal gland to secrete glucocorticoids, mainly cortisol. Cortisol is a type of glucocorticoid. Now, in turn, this elevated level of blood cortisol is going to decrease the secretion both of corticotropin, or adrenocorticotropic hormone, ACTH, and it's also going to decrease the secretion of the corticotropic releasing hormone by those hypothalamic neurosecretory cells. So increased levels of cortisol in the blood is going to suppress the activity of the anterior pituitary corticotrophs, and it's also going to suppress the activity of the hypothalamic neurosecretory cells. So this is a negative feedback loop that we see with the hormones of the anterior pituitary. Now, somatotrophs, these are going to be the most numerous cells in the anterior pituitary, and therefore the human growth hormone is going to be the most plentiful anterior pituitary hormone. Now, the main function of the human growth hormone is to promote synthesis and secretion of small protein hormones called insulin-like growth factors. So this is going to promote, we can write out this whole thing here, promote the synthesis and secretion of those insulin-like growth factors. Those IGFs. Now, in response to the human growth hormone, the cells in the liver, skeletal muscles, cartilage, bones, and some other tissues are going to secrete IGFs. Now, these IGFs may either enter the bloodstream from the liver, or they're going to act locally in other tissues as autocrine hormones or paracrine hormones. Now, the function of IGFs are here. We're going to write down 3 different functions of the insulin-like growth factors. So our first one is that the IGFs cause cells to grow and multiply by increasing the uptake of amino acids into cells and accelerating protein synthesis. So they're going to increase cell growth and division. by increasing uptake of amino acids. So that increase in uptake of amino acids is really going to accelerate protein synthesis. Now our second function is that the IGFs also enhance lipolysis in adipose tissue, and this results in an increased use of the release of fatty acids for ATP production by the cells of the body. So we're going to increase or enhance lipolysis. This is mostly going to be in adipose tissue where the most lipids are found, which is also going to increase use of fatty acids to make ATP. So they're really going to increase ATP production by using lipids more so. Another big function of IGFs. And then our last major function of those insulin-like growth factors is that in addition to affecting protein metabolism and lipid metabolism, the human growth hormone and insulin-like growth factors also influence carbohydrate metabolism by decreasing glucose uptake, which decreases the use of glucose for ATP production by most body cells. So they're kind of shifting which metabolism these cells use in order to create ATP. So instead of using carbohydrates for their main metabolism source, their IGFs are going to kind of shift cells into using more lipids and proteins for their metabolism. Now, somatotrophs in the anterior pituitary will actually release bursts of human growth hormone every few hours, especially during sleep. This is one reason why sleep is so important, especially for kids. In these large growth spurts, sleep is very important for that secretion of human growth hormone and insulin-like growth factors to overall help to repair and grow the body. Now, the secretion activity of somatotrophs is controlled mainly by two hypothalamic hormones. This is the growth hormone releasing hormone, also known as GHRH. This is going to promote the secretion of the human growth hormone. And the other hypothalamic hormone that controls the activity of somatotrophs is the growth hormone inhibiting hormone, GHIH. And this is going to suppress hormone secretion from somatotrophs. Now, GHRH and GHIH both have regulators themselves. So major regulators of the secretion of these two hormones is going to be blood glucose level. So if we have low blood glucose level, hypoglycemia, this is going to stimulate the hypothalamus to release GHRH, that growth hormone releasing hormone. And it's going to flow toward the anterior pituitary through those hypophyseal portal veins. And when it reaches the anterior pituitary, the GHRH is going to stimulate somatotrophs to release the human growth hormone. Now, that human growth hormone is going to stimulate the secretion of insulin-like growth factors, which speed up the breakdown of liver glycogen into glucose, which enters the blood more rapidly. So insulin-like growth factors help to break down that liver glycogen into glucose, and that glucose stays in the blood because remember that insulin-like growth factors also kind of restrict the cells from taking in glucose. So it's going to stay within the blood. Now, as a result, blood glucose level is going to rise to the normal level, about 90 milligrams per 100 milliliters. That's milligrams of glucose to milliliters of blood plasma. Then an increase of blood glucose level above the normal level, if it goes too high, then hyperglycemia is going to inhibit the release of any more GHRH. So that hyperglycemia, this is high blood glucose. So an abnormally high blood glucose concentration, this, on the other hand, is going to stimulate the hypothalamus to release GHIH. And remember, hyperglycemia also is going to inhibit the secretion of GHRH. Now, GHIH is also going to travel to the anterior pituitary by those hypophyseal veins, those portal veins. And GHIH is going to inhibit the secretion of the human growth hormone by those somatotrophs. So now human growth hormone is not being secreted, and so we get low levels of human growth hormone and also the insulin-like growth factors. And this is going to slow down the rate of glycogen breakdown, and so glucose is going to enter the blood more slowly. Then blood glucose levels are going to fall down back to normal, but if it falls too low, then we reach kind of a hypoglycemia level, and hypoglycemia is going to inhibit the release of GHIH and also stimulate the release of GHRH again. So we kind of have this continuous regulating cycle occurring always. So here we have the 7 hormones that the anterior pituitary gland secretes. So first is the human growth hormone, abbreviated HGH. This promotes the synthesis and secretion of the insulin-like growth factors. Remember, these are abbreviated IGFs. Next is the thyroid stimulating hormone, TSH. Now this is going to stimulate the synthesis and secretion of the 2 thyroid hormones. Remember, these are T3 and T4. Those are our 2 thyroid hormones. We will discuss those hormones a little bit later too. Next is the follicle stimulating hormone, FSH. This, in females, is going to initiate the development of several ovarian follicles and also stimulates those follicles to secrete estrogens. And in males, the FSH promotes sperm production. We also have the luteinizing hormone, LH. This triggers ovulation in females. Prolactin. This initiates and maintains milk production by the mammary glands. The adrenocorticotropic hormone, ACTH. This controls the production and secretion of cortisol and other glucocorticoids by the cortex of the adrenal glands. So remember, cortisol is a type of glucocorticoid that is secreted by the adrenal gland. And then lastly, the anterior pituitary also secretes melanin stimulating hormone, MSH, which doesn't really have a well-known role in humans, but we do know that in amphibians, this particular hormone increases pigmentation, which is where it got its name from, the melanocyte stimulating hormone. So this table here summarizes those principal actions of the anterior pituitary hormones. Essentially, we've gone over all these already, but a nice summary table is good to study for. Shows the hormones on this column, has the target tissues here, which is also important, and then those principal actions as well. So a nice table to use to help study. Now, though the posterior pituitary, also known as the neurohypophysis, doesn't synthesize its own hormones, it does store and release two hormones. Now, this part of the pituitary gland consists of axons and axon terminals of more than 10,000 hypothalamic neurosecretory cells. These cells secrete the two hormones oxytocin and antidiuretic hormone, or ADH. Now, the cell bodies of the neurosecretory cells are in the paraventricular and supraoptic nuclei of the hypothalamus, and their axons form the hypothalamo-hypophyseal tract. That's a tongue twister there, hypothalamo-hypophyseal tract. The hypothalamo-hypophyseal tract begins in the hypothalamus and it ends near blood capillaries in the posterior pituitary. The neuronal cell bodies of the paraventricular and supraoptic nuclei will synthesize the hormones oxytocin and antidiuretic hormone. You can see them just above here, above the optic chiasm here. Now, blood is supplied to the posterior pituitary by the inferior hypophyseal arteries, which branch from the internal carotid arteries. Now, in the posterior pituitary, the inferior hypophyseal arteries will drain into the capillary plexus of the infundibular process. This is a capillary network that receives the secreted oxytocin and antidiuretic hormone. So this capillary plexus here is the capillary plexus of the infundibular process. Now, from the plexus, the hormones will pass into the posterior hypophyseal veins here for distribution to the target cells and other tissues. So, as the name implies, antidiuretic hormone is a substance that decreases urine production. So, anti meaning against, dia means throughout, and uresis or uretic means urination. So, antidiuretic means a decrease in urine production. So antidiuretic hormone causes the kidneys to return more water to the blood, thus decreasing the urine volume. Now, in the absence of antidiuretic hormone, urine output will increase more than tenfold, from the normal 1 to 2 liters to about 20 liters a day. So this is when there's no antidiuretic hormone. You can urinate about 20 liters a day. Now, antidiuretic hormone also decreases the water lost through sweating, and it also causes constriction of the arterioles, which increases blood pressure. So what this means is that the amount of antidiuretic hormone secreted varies with blood osmotic pressure, and it also varies with blood volume. So an increase in blood volume causes a decrease in antidiuretic secretion, and the opposite is true as well. Decrease in blood volume causes an increase in antidiuretic secretion. So remember that antidiuretic hormone causes the kidneys to return more water to the blood. So this directly is going to increase blood volume. So bringing more water into the blood from the urine increases blood volume and also decreases urine volume. Antidiuretic hormone also decreases water loss through sweating. And it also causes constriction of arterioles, those small blood vessels. And this is going to directly cause an increase in blood pressure. Oops. How do we write that here? So constriction of arterioles increases blood pressure, but increasing the overall blood volume, this is also going to increase blood pressure too. So antidiuretic hormone directly causes two different pathways that both work to increase blood pressure. So really, really interesting here. So in this picture here, we have the regulation of ADH and also the actions of that antidiuretic hormone. Before we look at that though, I'm going to quickly differentiate some terms that we're going to see here. So first one is blood osmotic pressure. Now blood osmotic pressure is going to be the solute to liquid ratio in the blood. So what this means is that if you have a lot of solutes in your blood, then you're going to have a very high blood osmotic pressure. If your blood volume is very, very high, there's a lot of liquid in your blood, but not as much solute, your blood osmotic pressure will be low. Now, blood osmotic pressure is different from your regular blood pressure. Now, blood pressure is just the pressure of blood in the circulatory system. Now this is just the force of the entire fluid pushing against the walls of the blood vessels. So this is the pressure that they test when you go to the doctor's office with a blood pressure cuff. So this is just the pressure of blood pushing against the blood vessels. So these two terms are different. So if we have a high blood osmotic pressure, this could also be the same thing as a low blood volume. If we have low blood volume overall, the solute to liquid ratio is going to be pretty high, and so there's going to be a lot of solute within the liquid. Now if we add a lot of liquid but the solute doesn't really change, then our blood osmotic pressure will decrease, but the blood pressure itself will increase. So hopefully that kind of makes sense. We can kind of do it just with two little beakers here. So if we have two beakers, they're going to have the same amount of solutes, so they're both going to have 5 solutes. Now in this beaker we have low volume, but we have really high blood osmotic pressure. And if we were to put like a cap on it, our overall pressure would be low. Because the liquid's not pushing up against the cap or anything like that, it's very low pressure. Now if we were to have the same 5 solutes in here, the solutes are much spread further apart. The overall concentration of solutes within this fluid is quite low. So with this beaker, we're looking at a much higher volume. We have a low blood osmotic pressure because that solute to liquid ratio is much lower than in the other beaker, and our pressure has increased, our overall pressure. So example, if we put a cap here, now the water, the fluid is like pushing up against that cap, causing pressure within that beaker. So hopefully that kind of figures out the differences between blood osmotic pressure and blood pressure. So let's look at the regulation of antidiuretic hormone here. So on the left side here, we have a high blood osmotic pressure. So kind of like this beaker here. We have a lot of solute compared to a small amount of liquid. So this could also be a low blood volume. So this could be due to dehydration. It could be due to a decline in blood volume because of blood hemorrhaging. You're losing a lot of blood elsewhere. This also could be due to things like diarrhea or excessive sweating. Now, in this particular instance, it's going to stimulate those hypothalamic osmoreceptors. They're going to detect this high blood osmotic pressure. So remember those osmoreceptors, these are neurons in the hypothalamus that monitor blood osmotic pressure. So that's these osmoreceptors here. Osmoreceptors monitor blood osmotic pressure. So an elevated blood osmotic pressure activates these osmoreceptors directly. These osmoreceptors will also receive excitatory input from other brain areas when the blood volume decreases. So these osmoreceptors are kind of getting input from several different places. Now the osmoreceptors will activate the neurosecretory cells in the hypothalamus, these hypothalamic neurosecretory cells, and these are going to synthesize and release antidiuretic hormone. Now, when the neurosecretory cells receive this input from the osmoreceptors, they generate nerve impulses that causes exocytosis of this antidiuretic hormone containing vesicles from their axon terminals in the posterior pituitary. So that's kind of labeled here. Liberate that antidiuretic hormone from terminals in the posterior pituitary, and it releases those into the blood capillaries surrounding the posterior pituitary. Now the blood is going to carry ADH to 3 target tissues. So the kidneys, the sudoriferous glands, and the smooth muscle in blood vessel walls. So the arterioles. So this is what their responses are going to be when ADH is increased. So the kidneys respond by retaining more water. They're going to bring some of that water in the urine back into the bloodstream. They just kind of extract some water, put it back into blood, and this is going to decrease the urine output. The sweat glands, they're the sudoriferous glands, they're going to kind of respond by decreasing their secretory activity. So they're going to decrease their water loss and they're going to lower the rate of secretion, lower the rate of water loss by perspiration from the skin. Then the arterioles are also going to respond, or the smooth muscle in the walls of the arterioles are going to respond by contracting in response to these high levels of ADH. Now, when the smooth muscles contract, it constricts the blood vessels themselves. This says constricts here. And it narrows the lumen of these blood vessels, which increases blood pressure. So these are all three ways that increase blood volume and also increase blood pressure. So the smooth muscles don't actually increase the volume, but they will increase blood pressure. So that's how we kind of respond if we have low blood volume or high blood osmotic pressure. Now, on the other hand, if we have low blood osmotic pressure or we have really high blood volume, this is going to inhibit the osmoreceptors. It's going to inhibit the osmoreceptors, which reduces or stops that antidiuretic secretion of the hypothalamic neurosecretory cells. So in this case, when there's low levels of antidiuretic hormone being secreted, kind of stops these pathways here. So the kidneys then retain less water, so they can form a larger volume of urine. The secretory activity of the sweat glands increases, and the arterioles dilate. So in this way, then the blood volume and the osmotic pressure of body fluids is going to return back to normal. So here we have the two hormones that are secreted by the posterior pituitary. Recall that these are going to be hormones that are created by the hypothalamic neurosecretory cells, but that are secreted by the posterior pituitary. So this table lists those hormones, oxytocin and the antidiuretic hormone, which is also called vasopressin. It also lists the control of their secretion and their principal actions. Now, the secretion of antidiuretic hormone can also be altered in other ways, like pain or stress, even things like trauma, anxiety, alcohol and drugs, even morphine, just to name a few other ways that antidiuretic hormone can be controlled. Moving on to the thyroid gland. The thyroid gland is this butterfly-shaped gland located just inferior to the larynx. Now it's composed of a right lobe and a left lobe, and these are connected together with an isthmus. Now there are microscopic spherical sacs called thyroid follicles that make up most of the thyroid gland. Now the wall of each follicle consists primarily of cells which are called follicular cells. The follicular cells produce two hormones, thyroxine, which is also called T4, tetraiodothyronine, and this is because it contains 4 atoms of iodine. And the second hormone produced by the follicular cells is triiodothyronine, known as T3. And it's called T3 because it contains 3 atoms of iodine, or triiodo. Now, T3 and T4 together are known as the thyroid hormones. Now, there are a few cells called parafollicular cells, also known as C cells, that lie between the follicles. And they produce the hormone calcitonin, which helps to regulate calcium homeostasis. This is really important hormone for calcium homeostasis in the body. Now, the thyroid gland is the only endocrine gland that stores its secretory product in large quantities. Normally, they store about 100-day supply. So here we can see the location of the thyroid gland. So we can see it is in front of the trachea. It's just inferior to the larynx and then ventral to the trachea. Now in this right figure, we can also see the layout of the thyroid follicles. So we can see the entire follicle here. We can see those follicular cells lining the follicle and the parafollicular cells just outside of the follicles. So here on this slide, we have the synthesis and secretion of the thyroid hormones, T3 and T4. Now this entire process is about an 8-step process, and we'll go through the whole thing. So our step 1 here is down here in the bottom left, which is called iodine trapping. So this is where the thyroid follicular cells, which is this big yellow cell here, they trap iodine ions by actively transporting them from the blood down here into the cytosol. So you can see all these iodine molecules that they're actively transporting in. Now, because of this first simple process, the thyroid gland normally contains most of the iodine in the whole body because they're actively transporting all this iodine into the follicular cells. Our step 2 is going to be right here, and this is the synthesis of thyroglobulin. Thyroglobulin, also abbreviated TGB. So while the follicular cells are trapping iodine, at the same time, they're also going to be synthesizing thyroglobulin. So thyroglobulin is this large glycoprotein that is produced in the rough endoplasmic reticulum. It's going to be sent to and modified in the Golgi complex, and then it's packaged into secretory vesicles. Now these vesicles will then undergo exocytosis right here, which then releases the TGB molecules into the lumen of the follicle. So this is kind of within this space here inside the lumen of the thyroid follicle. So right now we're looking at these follicular cells that are sending the TGB into the center here. So that's going to be our step 2. Pretty much happening at the same time as step 1's happening though. Oops, then we have step 3. Go back to that blue. Step 3 is the oxidation of iodide. So this is going to be up here, but I'll write it out here too. So some of the amino acids in TGB are tyrosines, and they will become iodinated. So what this means is that the tyrosine is going to accept an iodine ion. However, these negatively charged iodine ions, these can't bind to tyrosine until they undergo oxidation. Remember that oxidation is a removal of electrons. So these iodide ions have to go through oxidation, that removal of electrons, and then they turn into iodine. So iodine, they have to be bound to each other. So instead of the I negative, that ion, they will become oxidized and turn into I2, which is going to be two iodine molecules bound to each other. So that happens in the lumen of the thyroid follicle. Now step 4 here is the iodination of tyrosine. Sorry, tyrosine is with an S here. So as these iodine molecules form, they're going to react with tyrosines that are part of those thyroglobulin molecules. Now binding of one iodine atom yields a monoiodotyrosine, which is T1. So binding of one iodine molecule is T1, and a second iodination produces diiodotyrosine. So binding a second iodine gives us T2, diiodotyrosine, and monoiodotyrosine. Now we have TGB with its attached iodine atoms. This turns into kind of a sticky material that accumulates, and it's stored in the lumen of a thyroid follicle, and it's called the colloid. So all those TGBs with their iodine molecules attached to them, either as monoiodotyrosine or diiodotyrosine, becomes kind of this sticky gelatinous material, and it's termed the colloid. You can see it here too. Now our fifth step is the coupling of T1 and T2. So during the last step in synthesis of the thyroid hormones, two T2 molecules will join to form T4, and one T1 and one T2 will join to form T3. So if we get T2 plus T2 is going to be a T4 molecule, and a T1 plus a T2 is going to equal a T3 molecule. So those are our two thyroid molecules. Our step 6 is pinocytosis and digestion of the colloid. So droplets of the colloid will actually re-enter the follicular cells by pinocytosis. So we can see this here. And they will merge with lysosomes. Now the digestive enzymes in the lysosomes will break down TGB and it cleaves off molecules of T3 and T4. Our 7th step is the secretion of the thyroid hormones here. So because T3 and T4 are lipid soluble, they will diffuse through the plasma membrane into the interstitial fluid and then into the blood. So they can diffuse rather quickly just straight into the blood because they are lipid soluble. So then our 8th step here is the transport in the blood. So T3 and T4, again, because they are lipid soluble, they combine with transport proteins in the blood. Our main transport protein that the thyroid hormones connect to is thyroxine binding globulin, which is also labeled down here, TBG. Not to be confused with the TGB of the colloid. So TBG transports the thyroid hormones in the bloodstream. Because most body cells have receptors for thyroid hormones, T3 and T4 exert their effects throughout the whole body. These are just some things that the thyroid hormones do throughout the body. So the first one is that they increase the basal metabolic rate, or your BMR. They also increase the rate of oxygen consumption under standard or basal conditions. A second major effect of thyroid hormones is that they stimulate synthesis of additional sodium-potassium ATPase pumps, which use large amounts of ATP to continually eject sodium ions from the cytosol into the extracellular fluid, and at the same time, they move potassium ions from the extracellular fluid into the cytosol. Now, as this is all happening, as cells produce and use more ATP, there's more heat given off, and so body temperature rises. So thyroid hormones increase the number of sodium-potassium ATPase pumps, and so as more ATP is made and used, it creates more heat. In the regulation of metabolism, the thyroid hormones stimulate protein synthesis, and they increase the use of glucose and fatty acids for ATP production. So they're increasing ATP production all around. The thyroid hormones enhance some of the actions of catecholamines. Catecholamines are epinephrine and norepinephrine. So epinephrine and norepinephrine are the two catecholamines. And the thyroid hormones enhance the actions of epinephrine and norepinephrine because they upregulate beta receptors on cells. And then also, together with the human growth hormone and the hormone insulin, thyroid hormones accelerate body growth, particularly the growth of the nervous and skeletal systems. The thyrotropin releasing hormone from the hypothalamus and the thyroid stimulating hormone from the anterior pituitary stimulate the synthesis and release of thyroid hormones. So here we have the 5 steps of that process. So when there are low levels of T3 and T4, or there's low metabolic rate, this is going to stimulate the hypothalamus here to secrete TRH. Remember, that's the thyrotropin releasing hormone. Now, thyrotropin releasing hormone enters the hypophyseal portal veins and it flows to the anterior pituitary. And it's here where it stimulates thyrotrophs to secrete thyroid stimulating hormone, TSH. Now, TSH will stimulate virtually all aspects of thyroid follicular cell activity. So it includes things like iodide trapping, as we saw in the prior figure with the follicular cells. It also promotes hormone synthesis and secretion, and also overall growth of the follicular cells. So TSH is going to really stimulate the thyroid follicle overall to produce more T3 and T4. So the thyroid follicular cells will release T3 and T4 into the blood until metabolic rate returns back to normal. Now, an elevated T3 will further inhibit the release of any more TRH and TSH. So we get that negative feedback when T3 levels are increased. Now this figure is cool here because it also has the actions of thyroid hormones down below. This is one that we talked about just a few slides ago. So it's also all listed here. This figure here shows the produced and secreted hormones from the follicular cells. So here at the top we have T3 and T4, the thyroid hormones secreted by the follicular cells. And it also shows the secretions from the parafollicular cells, namely calcitonin. It shows their control of secretion and their principal actions. Again, we've talked about all of these, but again, just a summary table here is helpful for studying. Now, partially embedded in the posterior surface of the lateral lobes of the thyroid gland are several of these small round masses of tissue called the parathyroid glands. So we can see them here. Here's one, here's one. These little small green colored masses in this picture. Now each of them has a mass of about 40 milligrams. Usually there's one superior and one inferior on each lobe of the thyroid, so total of 4 parathyroid glands. Now microscopically, the parathyroid glands contain 2 types of epithelial cells. The more numerous are called chief cells, also known as principal cells, and these produce the parathyroid hormone, also called PTH. Now the other cell are called oxyphil cells, And the function of the oxyphil cells is not really known in normal parathyroid glands, like in normal acting parathyroid glands. We do know what they do when the parathyroid contains cancer, but in normal acting parathyroid glands, the oxyphil cells function is not really known. However, the presence of oxyphil cells really helps to clearly identify parathyroid glands histologically, and this is due to its really unique staining characteristics. So here we can see those really nice staining characteristics of the oxyphil cells. This really helps to identify these parathyroid glands. So we can see these chief cells, they are small, very darkly colored, whereas the oxyphil cells are large, rounded, and they kind of have this cloudy stain. They're not as darkly stained as the chief cells. So this helps us to really identify parathyroid glands under the microscope. Now, the parathyroid hormone is a major regulator of levels of calcium, magnesium, and phosphate in the blood. The parathyroid hormone acts upon the kidneys as well. So it slows the rate at which calcium and magnesium are lost from the blood into the urine. Second thing that the parathyroid hormone does to the kidneys is that it increases the loss of phosphate from blood into the urine. And it also promotes the formation of the hormone calcitriol. Remember, that's the active form of vitamin D. Now, blood calcium level directly controls the secretion of both calcitonin and parathyroid hormone by negative feedback loops that don't really directly involve the pituitary gland. So this is what we're going to look at with a picture on the right here. So when we have a high level of calcium in the blood, this is going to stimulate the parathyroid or the parafollicular cells of the thyroid gland to release more calcitonin. All right, good. Calcitonin, abbreviated CT. So the calcitonin will actually inhibit the action of osteoclasts and thereby decreasing the blood calcium levels. So then we have a lower level of calcium in the blood, and if it goes too low, then it will stimulate the parathyroid gland chief cells to release more parathyroid hormone. Now the parathyroid hormone down here will then promote reabsorption of bone extracellular matrix, which is going to release calcium into the blood. And the parathyroid hormone will also slow the loss of calcium in the urine. Both of these actions overall increasing blood calcium levels. Now the parathyroid hormone is also going to stimulate the kidneys to synthesize calcitriol. that active form of vitamin D. And calcitriol will stimulate increased absorption of calcium from foods that are in the gastrointestinal tract that you eat. And this is also going to help to increase blood calcium levels. This is all just kind of this negative feedback loop, because then if we increase the blood calcium levels from the food due to calcitriol, and then we get too high, then we get back to step one over here, and then we have too high levels of blood calcium causing more secretion of calcitonin, and the cycle continues. This table here summarizes the control of secretion and the principal actions of the parathyroid hormone that's secreted from the chief cells of the parathyroid gland. Now next we're going to look at the adrenal glands. They are paired glands, one of each lies superior to each kidney in the retroperitoneal space. And they kind of have this flattened pyramidal shape. So you can see the left adrenal gland here on top of the left kidney, and the right adrenal gland here on top of the right kidney. Now in an adult, each adrenal gland has a mass about 3.5 to 5 grams, only half of its size at birth. Now during embryonic development, the adrenal glands will differentiate into two structurally distinct regions. There's a large peripherally located adrenal cortex. This makes up about 80 to 90% of the gland. And then there's a small centrally located adrenal medulla. Now there's a connective tissue capsule that covers the entire gland. And just like the thyroid gland, the adrenal glands are very highly vascularized. Now the adrenal cortex is subdivided into 3 zones. Each of these zones secretes different hormones. So we have the capsule, and the outermost zone, just deep to the connective tissue capsule, is the zona glomerulosa. Now its cells are very tightly packed together and arranged in small spherical clusters, and they secrete the hormone mineralocorticoids. And they're called mineralocorticoids because they affect mineral homeostasis. Now the main one is going to be aldosterone. The middle zone is the zona fasciculata. Now this is going to be the widest of the 3 zones, and it mostly consists of cells arranged in really long straight columns. Now these cells mainly secrete glucocorticoids, primarily cortisol. Then the cells of the innermost zone, which is the zona reticularis, these are arranged kind of in branching cords, and they synthesize small amounts of weak androgens. Androgens are steroid hormones that have masculinizing effects. Here, this slide pretty much says the same thing that I just talked about in the last slide, just in a word format. So it talks about the zona glomerulosa secreting mineralocorticoids, the zona fasciculata secreting glucocorticoids, and then the zona reticularis secreting androgens. Aldosterone is the major mineralocorticoid that is secreted by the adrenal gland. Now it regulates the homeostasis of 2 mineral ions, mainly sodium and potassium. So it helps to adjust blood pressure and blood volume. Now, aldosterone also promotes the excretion of hydrogen ions in the urine. Now, this removal of acids from the body can help prevent acidosis. Remember, that's a blood pH below 7.35. Now, a really important pathway controls the secretion of aldosterone. This is the renin-angiotensin-aldosterone pathway, also known as the RAA pathway. So this nice diagram here shows us the renin-angiotensin-aldosterone pathway. Yes, it's not particularly labeled here, but I'll put it here. So the renin-angiotensin aldosterone pathway, also known as the RAA pathway. So it looks a bit complicated, but I assure you it is not. We will go through it. So first off, we're just going to go through the numbers of the pathway, starting with number 1 in the top left corner here. So stimuli that initiate the RAA pathway include things like dehydration, sodium deficiency, or hemorrhage. Now, these conditions cause a decrease in blood volume, and decreased blood volume causes a decrease in blood pressure. Now, lowered blood pressure is going to stimulate certain cells of the kidneys. These are called the juxtaglomerular cells in the kidneys. Now, these cells are going to secrete the enzyme renin. So this will increase renin in the blood when these cells are stimulated by the decrease in blood pressure. Now, renin is going to convert angiotensinogen, which is a plasma protein produced by the liver, and renin is going to convert angiotensinogen into angiotensin 1. Now, angiotensin 1 is now an activated protein, and blood containing these increased levels of angiotensin 1 are going to circulate through the body. And as blood flows through capillaries, particularly those in the lungs, the enzyme angiotensin converting enzyme, also known as ACE, is going to convert angiotensin 1 into the hormone angiotensin 2. So ACE is this enzyme found in the lungs. So angiotensin 2 is a hormone, and as ACE converts angiotensin 1 to angiotensin 2, the blood levels of angiotensin 2 are going to increase. Now, angiotensin 2 stimulates the adrenal cortex in the adrenal gland to secrete aldosterone. And so we're going to get blood containing increased levels of aldosterone circulating to the kidneys, while circulating in the blood, and it will eventually reach the kidneys. Now on to number 12 here. In the kidneys, this aldosterone is going to increase the absorption of, or the reabsorption of sodium, which in turn is going to cause reabsorption of water by osmosis, because remember, water is going to follow salts like sodium. And as a result, then less water is lost in the urine. Now, aldosterone is also going to stimulate the kidneys to increase secretion of potassium and hydrogen ions into the urine. So we're getting rid of potassium and hydrogen, and we're holding on to sodium and water, keeping that in the body, in the blood. Now, with increased water absorption by the kidneys, our blood volume is going to increase. Then on to number 14 here. As blood volume increases, the blood pressure is also going to return back to normal. So this is kind of our end result. This is where we want to end up, with our blood pressure back to normal. Now there are a couple of other things that happen in this pathway too. As you can see, number 15 and 16 we have yet to talk about. So let's go with number 15 here, which is what angiotensin II also does. So angiotensin II acts on the adrenal cortex. We already talked about that. But angiotensin II will also stimulate contraction of smooth muscle in the walls of arterioles. So we're going to get a resulting vasoconstriction of arterioles, which increases blood pressure, and so it also helps to raise blood pressure back to normal. So we're also going to get that help in increasing blood pressure directly from angiotensin II. Now at number 16 here, besides angiotensin II acting on the adrenal cortex, a second stimulator of aldosterone secretion is an increase in the potassium concentration of blood, or also in interstitial fluid. So increased concentration of potassium in any extracellular fluid is going to stimulate aldosterone secretion in the adrenal cortex. Now a decrease in the blood potassium level has an opposite effect. So a low potassium blood level will inhibit the secretion of aldosterone from the adrenal cortex. So kind of a big pathway. It is kind of a lot to remember, but if you kind of just follow the steps, it's not too complicated. The glucocorticoids, which regulate metabolism and resistance to stress, includes cortisol, which is also called hydrocortisone. It also includes corticosterone and also cortisone. Now, of these three hormones secreted by the zona fasciculata, cortisol is the most abundant. So cortisol really accounts for about 95% of glucocorticoid activity. Now really the secretion of glucocorticoids again is regulated by negative feedback, just like all the other hormones that we've seen so far. Now these glucocorticoids have several effects. The first one we'll talk about is protein breakdown. So cortisol, we can just talk about cortisol because it's the most abundant of the glucocorticoids at 95%. It's mainly going to be the one that we see. So cortisol increases the rate of protein breakdown, particularly in muscle fibers. And so this increases the liberation of amino acids into the bloodstream. So we're breaking down proteins and putting that product, the amino acids, into the bloodstream. Glucose formation is another thing that glucocorticoids help to control. So when stimulated by glucocorticoids, cortisol, liver cells may convert certain amino acids or lactic acid into glucose. And glucose can be used mostly by neurons and even some other cells for ATP production. But neurons in particular will only use glucose for ATP production. So this one's really, really important here. Lipolysis will be our next function or next effect of cortisol. So glucocorticoids can stimulate lipolysis. Remember, lipolysis is the breakdown of triglycerides and release of fatty acids from adipose tissue into the blood. cortisol also helps with the resistance to stress. So they provide tissues with a ready source of ATP to combat a range of stresses. These kinds of stresses include exercise, fasting, fright, surgery, trauma, even disease. Glucocorticoids will also make blood vessels more sensitive to other hormones, so adding to that resistance to stress. They also have a role in inflammation. So they actually have anti-inflammatory effects. And this is because cortisol inhibits white blood cells that participate in inflammatory responses. Now, unfortunately, glucocorticoids will also retard tissue repair, and so as a result, they slow wound healing. Now, the last thing that they do is the depression of immune responses. So high doses of glucocorticoids depress immune responses, and actually it's for this reason that they're often used in organ transplants, so that your body doesn't quickly attack the transplanted organ, that you suppress the immune system so that your body can get used to the organ before it tries to kill it off. So as we know, the control of glucocorticoids secretion occurs by a typical negative feedback system. So if we have low levels of glucocorticoids in the blood, mainly cortisol, this is going to be detected by the neurosecretory cells in the hypothalamus. And this will cause those same cells to increase their corticotropin releasing hormone. And together, the corticotropin releasing hormone, CRH, along with that decreased cortisol, will promote the release of ACTH by the anterior pituitary. Now, the adrenocorticotropic hormone flows in the blood to the adrenal cortex, where it stimulates the glucocorticoid secretion. So then we have increased glucocorticoid levels in the blood. It will return us back to homeostasis. And if our glucocorticoid level gets too high, then this would all cease this pathway, having that negative feedback. In both males and females, the adrenal cortex secretes small amounts of weak androgens. Now, the major androgen secreted by the adrenal gland is dihydroepiandrosterone, much easierly just pronounced DHEA. Now, after puberty in males, the androgen testosterone is also released in much greater quantities by the testes. So after puberty, the DHEA effect are quite insignificant in males. In females, however, adrenal androgens do play important roles. They promote libido or sex drive, and they're converted into estrogens, which are feminizing sex steroids by other body tissues. Now, after menopause, when ovarian secretion of estrogen ceases, all female estrogens come from the conversion of adrenal androgens. Now, the inner region of the adrenal gland is the adrenal medulla. And this is a modified sympathetic ganglion of the autonomic nervous system. Now, rather than releasing a neurotransmitter, the cells of the adrenal medulla secrete hormones. Hormone-secreting cells, called the chromaffin cells, These are innervated by sympathetic preganglionic neurons of the autonomic nervous system. Now, the two major hormones that are synthesized by the adrenal medulla are epinephrine and norepinephrine. These are also called adrenaline and noradrenaline. Now, the chromaffin cells of the adrenal medulla secrete an unequal amount of these hormones. Normally, it's about 80% epinephrine and about 20% norepinephrine. Both these hormones, though, are involved in the fight or flight response. In stressful situations and during exercise, impulses from the hypothalamus stimulate the sympathetic preganglionic neurons, which in turn will stimulate chromaffin cells to secrete epinephrine and norepinephrine. Now, this table here summarizes the hormones produced by the adrenal glands. So we have the adrenal cortex hormones, mineralocorticoids, glucocorticoids, and then we also have androgens. And then there's also the adrenal medulla hormones, epinephrine and norepinephrine. It also shows their control of secretion here and the principal actions. So again, we've gone over all this, but here's a nice summarizing table of all those hormones secreted by the adrenal gland. Next, we're going to be moving on to the pancreas. Now, the pancreas is both an endocrine and an exocrine gland. It's located in the curve of the duodenum, which is the first part of the small intestine, and it consists of a head, a body, and a tail. Roughly 99% of the exocrine cells of the pancreas are located in clusters called acinae. Now, the acinae produce digestive enzymes, which flow into the gastrointestinal tract through a network of ducts. Scattered among the acinae are about 1 to 2 million tiny clusters of endocrine tissue, which are called pancreatic islets. These are also called islets of Langerhans. So the acinae, these are going to be exocrine cells, and the pancreatic islets are endocrine tissue. Now, there's really abundant capillaries that serve both the exocrine and endocrine portions of the pancreas. So here we can see the acinae here, and then we can also see the pancreatic islet, which is pretty much this entire interior portion here. So we can see this. This is just the pancreatic islet with the surrounding acinae. So we can see how the acinae completely surround the pancreatic islet. Now, each pancreatic islet includes 4 types of hormone secreting cells. Now, the alpha or A cells, these constitute about 17% of the pancreatic islet cells, and they secrete glucagon. Beta cells, or B cells, constitute the largest percentage at about 70% of the pancreatic islet cells, and they secrete insulin. Delta cells, or D cells, these constitute only about 7% of the islet cells, and they secrete somatostatin. The F cells constitute the remainder of the pancreatic islet cells, and they secrete pancreatic polypeptides. Now, the interactions of these four pancreatic hormones is really quite complex, and it's actually not even completely understood. We do know that glucagon raises blood glucose level. And we know that insulin lowers blood glucose level. Somatostatin acts in a paracrine manner to inhibit both insulin and glucagon release from neighboring beta and alpha cells. So inhibits that release of glucagon and insulin. And the pancreatic polypeptide inhibits somatostatin secretion. So this is going to inhibit somatostatin there. This table here summarizes the hormones that are produced by the pancreas. So these are the pancreatic islet hormones. It also shows their control of secretion and also the principal actions. So on this slide, we have the alpha cells and beta cells. So again, these are secreting glucagon from the alpha cell and insulin from the beta cell. And then on this slide, we have the delta cell secreting somatostatin and the F cells secreting pancreatic polypeptides. Now, the principal action of glucagon, remember we talked about, is to increase blood glucose level when it falls below normal. Now, insulin, on the other hand, helps to lower blood glucose level when it's too high. So the level of blood glucose controls secretion of glucagon and insulin by negative feedback. So if we have a low blood glucose level, hypoglycemia, this is going to stimulate the secretion of glucagon from those alpha cells in the pancreas. Now, glucagon is going to act upon those hepatocytes, which are liver cells, and it acts on them to do a couple different things. To accelerate the conversion of glycogen into glucose. So again, this process here is called glycogenolysis. And glucagon also helps to promote the formation of glucose from lactic acid and also from certain amino acids. Now this bottom one here, this is a process called gluconeogenesis. These are terms I'm just kind of introducing now, but we'll definitely talk about these a lot more when we look at our metabolism chapter. These will definitely come back up. So as a result of these actions that glucagon has on the hepatocytes will release glucose into the blood more rapidly. And as a result, then blood glucose levels will rise. Now, if blood glucose continues to rise, then high blood glucose level, hyperglycemia, is going to inhibit the release of glucagon. So we get this negative feedback loop coming back up. And then coming back up and inhibiting that release of glucagon from the alpha cells. Now on the other side of this figure, we have hyperglycemia, so high blood glucose over here. So this is going to stimulate the secretion of insulin from those beta cells of the pancreatic islets. Now, insulin really acts on a lot of different cells of the body, but it works to do a couple things as well. So we're going to accelerate the facilitated diffusion of glucose into the cells. So it's going to help to remove glucose from the blood and put it into the cells. It's going to speed up the conversion of glucose into glycogen. This process is called glycogenesis. It also works to increase the uptake of amino acids, then also increase protein synthesis. And it also works to speed up the synthesis of fatty acids. So this is lipogenesis. Genesis, the creation of lipids. So as a result of these things, the blood glucose level is going to fall. And if blood glucose levels fall below normal, then low blood glucose levels is going to inhibit the release of any more insulin. So again, having that negative feedback loop. Now, hypoglycemia that we get kind of down here, that hypoglycemia is also going to stimulate those alpha cells to secrete glucagon. And hyperglycemia that we would get down here would also then stimulate the beta cells to secrete insulin. So kind of two separate but connected pathways here. Now the gonads are the organs that produce gametes. Gametes are sperm in males and oocytes in females. Now, in addition to their reproductive function, the gonads secrete hormones. So the ovaries, which are paired oval bodies located in the female pelvic cavity, produce several steroid hormones, including two estrogens, which are estradiol and estrone, and they also secrete progesterone. Now, these female sex hormones, along with the follicle stimulating hormone and luteinizing hormone from the anterior pituitary, these all work to regulate the menstrual cycle, maintain pregnancy, and also prepare the mammary glands for lactation. Now, the ovaries also produce inhibin and relaxin. Now, inhibin is a protein hormone that inhibits the secretion of follicle-stimulating hormone. So we'll say inhibits follicle-stimulating hormone. If you just kind of draw a line up to another thing, this is going to be kind of an inhibitory action. Now, during pregnancy, the ovaries and placenta produce a peptide hormone called relaxin. Now, relaxin increases the flexibility of the pubic symphysis during pregnancy and also helps to dilate the uterine cervix during labor and delivery. So it increases the increased flexibility of the pubic symphysis. Now the male gonads, the testes, are oval glands that lie in the scrotum. Now the main hormone produced and secreted by the testes is testosterone. Now testosterone, remember, is an androgen. Androgen is a male sex hormone. Testosterone stimulates the dropping of the testes before birth. It also works to regulate the production of sperm and stimulates the development and maintenance of male secondary sex characteristics, such as beard growth or deepening of the voice. Now we will discuss these particular hormones with respect to the reproductive systems. We'll talk a lot more in detail with them when we talk about the reproductive system chapter. So this table here just summarizes the hormones that are produced by the ovaries and by the testes. And it also says their principal actions here. Next, we have the pineal gland. Now this is a small endocrine gland that's attached to the roof of the third ventricle of the brain at the midline. Now the gland consists of masses of neuroglia and secretory cells called pinealocytes. So these are going to be the main secretory cells within that pineal gland. Now the pineal gland secretes melatonin. This is an amine hormone derived from serotonin. Melatonin appears to contribute to the setting of the body's biological clock. And remember that biological clock is controlled by the suprachiasmatic nucleus of the hypothalamus. Then we also have the thymus. This is located behind the sternum between the lungs, and it also produces several hormones. So here we have the hormones produced by the thymus. We have thymosin, the thymic humoral factor, THF, thymic factor, TF, and also thymopoietin. Now all of these hormones promote the maturation of T cells. Now T cells are a type of white blood cell that destroys microbes and foreign substances. So all we need to know for now is that is a type of white blood cell. So here in this picture, we can see the pineal gland right here, attaching to the roof of the third ventricle of the brain at the midline. And here we can also see the thymus, lying right behind the sternum in between the lungs. Now it's impossible to remove all the stress from our everyday lives. Now some stress is called eustress, and this actually prepares us to meet certain challenges, and so it's kind of a helpful type of stress. Now other stress, called distress, this is harmful stress. Now any stimulus that produces a stress response is called a stressor. Now your body's homeostatic mechanisms attempt to counteract stress. So when they're in a stressful environment, the internal environment remains within normal physiological limits, so maintaining homeostasis even through stress. But if the stress is extreme, unusual, or particularly long-lasting, the normal mechanisms may not be enough. Now, a variety of stressful conditions or noxious agents will elicit a similar sequence of bodily changes. Now, these changes are called the stress response. It can also be called general adaptation system, or general adaptation syndrome, GAS. And the stress response is really controlled mainly by the hypothalamus. The stress response occurs in 3 stages. So our initial fight or flight response is our stage 1. Stage 2 is a slower resistance reaction. And eventually, kind of the final stage of the stress response is exhaustion. Now our fight or flight response, this is really going to stimulate the body for immediate activity. The resistance reaction, this is going to be the second stage, and it lasts quite a bit longer than the first response. So one thing that happens with the stress response is that the stress response brings huge amounts of glucose and oxygen to the organs that are most active in warding off danger. So this would be the brain that becomes highly alert, also the skeletal muscles, which may have to fight off an attacker or flee, and the heart as well, which must work vigorously to pump enough blood to the brain and the muscles. So this is a diagram of the stress response. So we have stressors here that will stimulate the hypothalamus. Now here, the red arrows indicate hormonal responses, and the green arrows indicate neural responses. And then the black arrows indicate long-term resistance responses, or resistance reactions. So here we have this really short-lived fight or flight response, which is initiated by nerve impulses here. So these nerve impulses from the hypothalamus will initiate that short-lived fight or flight response. We can see this here affecting the sympathetic nerves and the visceral effectors here. These stress responses is all essentially the fight or flight response. Now, on the other hand, the resistance reaction is initiated in large part by the hypothalamic releasing hormones up here. And it is a much longer lasting response. So we see these releasing hormones affect their respective hormones in the anterior pituitary, which will then influence their respective organs. So the adrenocorticotropic hormone affecting the adrenal cortex to release cortisol. And we've talked about the stress responses of cortisol. The human growth hormone affecting the liver to secrete insulin-like growth factors, having these responses. And the thyroid stimulating hormone, obviously stimulating the thyroid to secrete thyroid hormones T3 and T4. And we already discussed that stress response as well. Or those, we didn't discuss these hormones specifically in stress responses, but we did discuss what cortisol, insulin-like growth factors, and the thyroid hormones do. And so specifically with regards to stress responses, this is how these hormones react. So this much longer lasting hormone-driven reaction is going to be that resistance reaction, the black arrows. Now, although some endocrine glands actually shrink as we get older, their performance may or may not be compromised. production of the human growth hormone by the anterior pituitary does decrease, which is one cause for muscular atrophy as aging proceeds. So some differences with aging in the endocrine system, but there's also a lot of ways where it doesn't change, even if there is anatomical changes to the endocrine glands. It may not cause physiological changes to the secretion of the hormones. So just like the other systems, we want to focus on homeostasis and how the endocrine system contributes to homeostasis of all the other systems. Hormones affect every other system. We've kind of talked about this kind of individually throughout the entire chapter, so just take time to study this and just kind of connect all the dots between the different organ systems that we've talked about with relation to the endocrine system, and essentially how they're all really interconnected. So take some time on this slide here. And that will be the end of chapter 16.